Hi everyone, it's Adam, the geneticist from Verdure Healthcare, and welcome to my brand new series, Genes That Can Make Your Baby Sick. So I'm doing this series for a free genetic education for everybody. So I'm going to talk about in today's introductory video on what is genetic disease, why does it affect healthy people like you and me, and how will they be passed on such bad genes to our next generations. So in the introduction, I will be having two parts. So the first one would be the bare basics, all you need to know about genetics, and the second part, which I'll be doing next week, will be on more advanced topics. And furthermore, in the future series, I'll be talking about genetic diseases one by one. I'll start off with the most common diseases in Malaysia. Right, so without further ado, you might be asking us, why do we need to get ourselves tested before getting pregnant? Well, in order to answer your question, I would like to provide you a study. Right, so it's a population-based study about birth defects in Malaysia, and this study is associated with the National Library of Medicine. So, honestly speaking, this is not a very happy ending kind of like study, because it is mentioned that 1 in 70 babies in Malaysia are born with either some kind of birth defects or genetic diseases. And it is my mission and also my vision to reduce this number in our country. But you might have a second question for me. Adam, I look healthy, right? My family is healthy, nobody has any genetic diseases, so what does that got to do with me? Well then, if we have a look at this research back in 2015, it is done by a very famous University of Chicago Medical School, along with Columbia University. And have a look at what it says. It's saying that humans will carry 1 to 2 lethal recessive mutation on average, study estimates. Okay, let me translate that into human language. It is saying that people, healthy people like you and me, we will be carrying at least 1 to 2 bad genes that can cause a disease if our spouse has it too. So let's have a look into it. So once again, humans carry on average 1 to 2 mutations. That's what it's called. Bad genes are called mutations. So if inherited from both parents, for example, if your spouse has it and you have it, can cause severe genetic disorder or death before reaching reproductive age. And now the so-called reproductive age would be around 15 years old, not 18. 18 is just the legal adult age, but scientifically speaking, reproductive age is around 15 years old. So this is quite a scary research that we have to face, right? So everybody has genetic mutations, including healthy people like me and you. So there is this, bad genes in us that is just lurking around in our body, and because it doesn't have enough power to cause literally a disease. So let me ask you this question and you will immediately understand what I'm trying to say. Okay, if you have a friend who's a couple and they come to you and say, oh, we're gonna have a baby, but for some reason, you know that these two, okay, this couple, they both have thalassemia genes. Thalassemia is a kind of blood disease, okay, the lack of blood, okay? So would you suggest them to have a baby? Well, maybe you're not going to say like, oh, since both of you are having alpha thalassemia genes, you're not, it's not advisable for you to have a baby. Maybe you wouldn't say that up in front of them, but deep in your mind, you will raise an alarm. So this is exactly what this study wants us to be aware of. Right, so what exactly are genes? Now you're looking at it, okay? So what you're looking at in here is like a twisted ladder, some kind of a shape, right? But each rung or each level on this twisted ladder will contain genetic information about us. So basically, this is an instructional manual. So it's guiding the body, our body, how to make the components, for example, like tissues and organs, things like that. So, well, I mean, this shape is all around us. There are some very clever people making such shapes into jewelries. I'm not sure whether you've seen them or even wearing one of them. And there are people saying, oh, even cars have DNA, like auto DNA, there's DNA in your BMW, things like that. And even there are computer DNA. So we are quite familiar with these terms. By the way, DNA and genes, they mean the same thing, right? And there are even some really smart people who can use malwares, meaning the viruses, turn them into DNA codes and hack into other people's computers. Wow, that is very smart. So what I'm trying to say is that more or less you will hear about DNA. And when you're not very familiar with it, you'll be confused. So let me clear up the confusion for you. So coming back to humans, okay, where exactly are our DNA? Okay, the most straightforward answer that you can think of course, would be the sperm cells and the egg cells. That's how we inherit things to our offsprings, right? To our children. But not many people know that even our motor neurons, our bone cells, our intestinal cells, our white blood cells, in fact, every single cell in our body will contain our DNA. Right. So, if we were to cut a cell into half, it pretty much looks like something from the outer space, isn't it? It looks like Star Wars. But anyway, all we need to focus on is this yellowish golf ball structure. So it is the command center in the cell, right? So all of these actions are being commanded by this command center. And so what does this command center include? Okay, it has our chromosomes. And chromosome and DNA, they are actually the same thing, right? But it is just the level of description that is different because DNA has been packed into chromosomes. So here is the clearer picture. As you can see, this is the cell and within it the command center, and the proper name for the command center within the cell is called the nucleus. Okay? So within this nucleus there are chromosomes. Chromosomes, if you unpack it, you will see the genes. And DNA and the genes, as I mentioned before, they are exactly the same thing. So a good example would be if you go to DIY shops, if you buy a ball of strings, right, it will be somehow like this shape. So it is more or less like a chromosome. And when you unpack this ball of strings, okay, it is like this chromosome unpacking their genes. So next time when you buy a ball of strings to tie something, you can think of it as though chromosomes are unpacking the DNA. Right, so let me tell you something interesting. Every life form, whether it is a plant, whether it's an animal, whether it's a human, they are made of the same DNA. Right, what do I mean by that? Okay, let's have a look at this deep level. So, there are only 4 ingredients, okay, in terms of DNA. There are only 4 kinds, G, C, A, and T. Now you don't have to remember them, but all you need to know is that there are only 4 types of DNA, G, C, A, and T. Well, so that's why we as humans, we share 60% similarity with a banana, right? Okay, many people don't know that. Okay, and if you compare a mouse gene and a human gene, we have 90% commonalities, right? And the differences in terms of DNA between each human, even if you compare a lady and a gentleman, they have 99.9% similar DNA. But what makes us, this visible difference? Okay, it is of course how you read this DNA, all right? And how much GCATs are in there. In humans, we have approximately 3.1 billion of these GCATs. So if you line them up from the floor all the way up, it would go almost to the outer space. That's how long our DNAs are. Anyway, so when we come back to diseases, so how many types of genetic diseases are there? Okay, I will boil it down to two types, okay? Single gene disorder, meaning to say that everything is perfect, everything else is perfect in you, but there is just something wrong with one of the GCATs that can cause you to have muscular dystrophies, okay, spinal atrophy, alpha thalassemia. All of these are just because one of these little, codes has gone wrong. All right, and on the second level, chromosomal abnormalities. Okay, so on this level, on the chromosome level, there could be, like large chunks being missing. Okay, you can even have an extra piece of chromosome, something like that. For example, Down syndrome. All right, which I'm going to explain right away. So this is our blueprint. In every single one healthy person, we must have 23 pairs of chromosome, right? So from the first pair all the way until the 22nd, they must be symmetrical. So even though one of those, one half comes from mom and the other one comes from dad, even their patterns have to be the same, all right? The only exception would be this, the sex chromosome. So if you are exactly like this, an X and a Y, then you are a guy. But if you don't have a Y chromosome, you have two X chromosome, then you are a lady. Right, so yeah, this is exactly the female version of the blueprint. So as you can see, if there are ladies listening to this Facebook Live right now, if you are lining up your chromosomes, this is exactly what you will be seeing. So in terms of chromosomal abnormalities in this category, how many kinds of things that can go wrong? So there are two types. Numerical abnormalities and structural abnormalities. Let me explain to you what I mean by that. For example, Down syndrome, okay? Everything else is perfect, except for the 21st pair of chromosome, there is an extra piece, right? And that's what it calls Down syndrome. I'm sure more or less in life you have come across a person or a baby with Down syndrome before. And there are circumstances whereby it is worse than trisomy, the Down syndrome. For example, this is called the Patau syndrome, and it is a very lethal disease whereby on the 13th pair of chromosome, there is an extra piece. And on the other hand, similar to Patau syndrome, the one before, this is called the Edwards syndrome, whereby on the 18th pair, there is an extra piece. So under this umbrella, this is called trisomy, okay? Because tri means three, trisomy meaning to say that there are three chromosomes. But do be aware that no matter if it is a Down syndrome, Edward or Patau syndrome, only one pair of chromosome has gone wrong. The other ones are absolutely perfect. Right, so on this extreme end, okay, whereby this is called Triploidy, whereby every slot of these chromosomes has an extra piece. Everyone has an extra piece. So this is an extremely lethal kind of condition, and most probably this baby will not be even born. So that's why we do have something called the non-invasive prenatal test. As early as the 10th week of pregnancy, we will be able to detect whether the fetus in mommy's tummy, whether the baby is healthy or not. We'll be able to look at all of these chromosomes, right? So if we were to go back to the previous slide, okay, some people have asked me, okay, Adam, all right, you told me that, okay, 18, 13, and 21, there can be something wrong, and these are the most common conditions, but what about the first pair of chromosome? What about 20th pair? Can they have trisomies as well? Oh yes, they do. They can have trisomies in all of these chromosomes. So it's a possibility from the first all the way until the 22nd pair. Although, for example, the 7th pair of chromosome having an extra piece is very rare, but we still have such conditions before. So that is why when we check your chromosomes, then we'll be able to see every single one of those. Right. So another condition, related to the numbers of chromosome is monosomy. So since conditions like having an extra piece can happen, how about having one less copy? Yes, it can happen. For example, with the 7th pair, there's one piece missing, and this is also a very lethal condition. Right, and one of the things that I want you to be aware of would be the sex chromosome abnormalities, whereby, for a normal male, you get an X and a Y chromosome. But what happens if you get an extra piece of an X chromosome? Or what happens if you get an extra piece of Y chromosome? Or if you have an X and a Y chromosome, but there are pieces missing in the Y chromosome. So all of these are related to the fertility, right? So if, let's say, for example, this one, Y chromosome rearrangements, or what we call deletion, meaning to say bits and pieces missing, it will definitely affect male fertility. Previously, we had a client whereby his Y chromosome had like 50% missing. Then what happened was that he has got azoospermia. Azoospermia meaning to say that he has got no sperm. He couldn't produce any sperm cells at all. Right, so on the lady side, if you're a healthy lady, you get an X chromosome and another X chromosome. But there are conditions such as Turner syndrome, whereby either you have the entire piece of X missing, some bits of X is missing, or have an extra piece of X chromosome, and this is called trisomy X syndrome. So just like the male counterpart, what this lady will be facing is female infertility. For example, the ovaries are not growing, not ovulating, or even having the lack of reproductive organs. So according to some studies, Turner syndrome can lead to such conditions. Right. So let's talk about the structural abnormalities, because just now we talked about the numerical abnormalities. Now, how about the structure? If you don't have an extra piece, or in terms of numbers, you're normal and you wouldn't be having one less chromosomes, but there are 5 conditions: deletion, duplication, inversion, substitution, and translocation, that if you pass these on to your next generation will cause some not so happy events. And later on, after I finish introducing you to all of these, I will tell you a real case that we, Verdure Healthcare, has faced before. So let's start off with deletion. For example, this is a healthy chromosome. But if you have this piece missing, oh dear, then where is this piece gone? It's just gone, and then your body wouldn't get any information from this. And that's why it's a lethal condition as well. Right, duplication, meaning to say that, within the same, for example, just that we talked about deletion. So with the same region, chromosomal region, there is a duplication. So there is an extra piece of information within that chromosome. So this one and just now, the deletion, are both lethal conditions because the amount of genetic information is not correct. Right, and inversion, meaning to say that, within the same region, for example, there's a flip, and then this piece has actually gone to the other one, So it basically means flipped and swapped, flipped and swapped. So in terms of genetic information, there isn't any change at all. So for you, if you have such condition, you will be okay. Right, substitution means the same thing, except sometimes it might not be swapping between the same chromosome. So for example, the 4th chromosome and the 20th chromosome, there has been a swap. Okay, yes, it can happen. It can happen. Believe me, we've came up with this before. Translocation is almost the same thing. Right. So now, in terms of structural abnormalities, okay, like I said before, the deletion and duplication would be lethal because the amount of genetic information is not correct. So, what to do? Well, how can we, detect all of these? So we can't even see chromosomes with our bare eyes. Okay, so at Verdure Healthcare, we have something called the pre-pregnancy DNA screening. So there are two parts to it. So the first part is to check all of your chromosomes first. So on this level, we would confirm, okay, if the amount of chromosomes are correct. Are there any pieces being missing? Are there any information being swapped around? Now, here's the real case I want to show you. So this is one of our live reports, okay? So this test, okay, it says abnormal chromosome result. But what's wrong with it? Okay, 46 chromosome, 23 pairs, that's 46, and so no problem. XY, so this is a guy, okay, it's a husband in fact. So, but next one, T bracket 2 semicolon 6. Okay, then we know that there is something wrong with the second chromosome and the sixth chromosome. And let's have a look at what's wrong. Bracket P13 semicolon Q30. Okay, combine these two information together, so we now know that something from the second chromosome, P13, this is the location of the chromosome, has been swapped with the sixth chromosome on the Q13 location. So, what condition is this? Okay, let me know in the description box, okay? Right, so this is, the balanced reciprocal translocation. So this is, in fact, if we go back to the slide, this is exactly what happened. But, okay, let me test your knowledge a little bit. Will this person have anything wrong at all? Okay, if you are seeing this person, do you think that he will be a special person? Right? The answer is no. Why? Because, once again, okay, I know this is difficult, but there is no gain or loss of genetic information. So now this is the danger. If you don't have anything wrong, would you go and check your chromosome, out of nothing at all? You wouldn't, right? Not until if you want to know that, okay, your genes are healthy so that you don't pass on any bad genes to your offspring. Okay. But this is a very, dangerous condition. So first of all, why did this couple come to us? It is because his wife miscarried 7 times, right? 7 times miscarriage. This is very serious. So he came to us, and I organized the test for him, and then immediately I can see why did his wife miscarry 7 times. So his wife was completely normal. There's nothing wrong with her chromosomes. But for the husband, since number 2 and number 6 chromosomes genetic material has swapped, let's have a look at what can happen. So this is the probability of having an unhealthy child. So if this is the condition, okay, there will be 25%, meaning a quarter chance that this child will be healthy, all right? So there will be nothing wrong with the child at all. So balanced reciprocal carrier, there's a 25% chance, so just like his dad, whereby there's no gain or loss of genetic information, so all in all, the child would look healthy. But if he goes on to get married, and then he goes on to, have babies, then this risk will carry on. But there is half a chance whereby there will be an unbalanced translocation, or unbalanced duplication. So that's why we as geneticists, we need to do this kind of an assessment. Okay, we need to do this risk management so that we know the chances of having an unhealthy baby. And for this couple would be 50%. So now can you see the danger? 50%, are you going to gamble with it? Right? I'm sure majority of you will say no, that is just way out of our tolerance level. So in that case, what do we need to do? Okay, one way, first of all, how did we deal with this couple? We have to send them off to IVF. So first of all, we enhance the husband's sperm cells. Okay, we enhance it by the quantity, the quality, the swimming capabilities, and all that kind of parameters. And for the wife, we enhanced her ovulation functions, her egg qualities, and all of those related to female fertility. So we send them off to IVF, we enhance we boost their fertility just because we want their chance of success to be higher. And they have to go through something called the pre-implantation genetic screening, or PGS. Now, it's useful to remember this term, PGS, because that is one of the procedures your doctors might ask you whether you want to opt in or not. PGS. So PGS is like when the embryo is formed, they will take a piece of the cells out of this embryo and they will perform the chromosomal screening for the embryo. That is PGS. Right. So yes, right. Okay, I want you to take a break after this. Okay, so that comes to the conclusion of chromosomal abnormalities. But last but not least, the word of the day would be cytogenetics. Okay, so cytogenetics is basically the study of chromosomes. So all of the things that I just talked about, including duplication, trisomy, monosomy, Turner syndrome, all of these belong to cytogenetics. And in Chinese, we call this 细胞遗传学. So if you're interested, you might want to have a look at it. But all in all, if you fully understand what I've just said, everything about chromosomes, you are already a cytogenetics expert. Right, okay, so now the thing is that analyzing a cytogenetic report is not the most difficult part. The more difficult part is to assess the risk. If, for example, like just now, the couple has balanced reciprocal translocation, you need to do the risk assessment. So you need to come up with a viable fertility solution. So that's the most difficult part. And this part should be done by qualified geneticists. So for example, I myself, I'm a Stanford certified genetic expert. I hold the Stanford Genetics and Genomics Certificate. But that still doesn't mean that I can analyze cytogenetic report. After this, I still have to go with the KKM, Malaysian Ministry of Health, their training in conventional cytogenetic. Okay, only then you are qualified to analyze and to produce fertility solution and risk assessment for any chromosomal reports. So that's why in the market or in the industry, there aren't many cytogenetic experts. That's why, because the training is really quite rigorous and the training could be quite difficult at times. Okay, right. So in the part 2, okay, remember we have 2 parts of this introductory video. In the part 2 of it, I will talk about the 155 most common genetic diseases in Asia, or in Asia Pacific to be a bit broader, okay, that is caused by single gene. Just this one itsy bitsy gene that is enough to cause horrible diseases like spinal muscular dystrophy, something like that. And anyway, I want you to take a break. And if you have time, you can go through this video again. And if you decide to have a baby, please give us a call. We would love to hear from you. And I hope you benefit from this video. And after this Facebook premiere, this video will be put on YouTube. Okay, this video will be put on YouTube. I want you to go on to the channel and watch this video again, and I hope you will like us, share this video, and subscribe to our channel so that you will get the first-hand news about our updates and also the next time when I talk about another kind of disease. Thank you very much. I wish you a great day ahead.